Hello, and I appreciate you attending my 2020 update on tick-borne diseases. I'm Holly Ahern, and I'm Associate Professor of Microbiology at SUNY Adirondack, and also a member of the advocacy, education, and research community when it comes to mostly Lyme disease. I think my greatest role here, my most important title, is that of mom, because it was as a mom that my adventure started with Lyme and tick-borne diseases, mostly because my daughter developed a very complicated case of Lyme disease. And what we learned in that process brought me to the Lyme world and to research and to advocacy today. So I'm going to, my objectives are to kind of give you some ideas of what ticks are important in our area, the diseases that they can carry so that you know what your risks are, show you where you can access some data, find data for yourself to find out what ticks are where and what those ticks are carrying, give you a little bit of background on symptoms and what tick-borne disease is in general, and then kind of focus on Lyme disease because of all of the tick-borne diseases, 85% of the cases are actually Lyme, and that's particularly true in our area. Also because Lyme has a disputed history in terms of what the disease is and what it can be. So I just want to give you some background on that, and I want to finish up with an update on the research on progress that we're making, that we are changing the landscape for Lyme disease, and it will get better from this point on. So regarding ticks, these are the ticks in New York State that have been found. Not that others have not been, but these are the most common types of ticks. In our area, we have the black-legged tick, which the scientific name for that, which is why it's written in italics, is Exodes. Exody Scapularis is the kind of tick that transmits Lyme disease, but it also transmits another type of Borrelia infection called Borrelia miyamotoi disease, which is being abbreviated BMD. And there is a growing consensus that a lot of cases of undiagnosed Lyme disease may actually be this related bacteria, which is Borrelia miyamotoi, because they cause pretty much the same symptoms, only Borrelia miyamotoi is not associated with a rash. There's also Babesia, which is a parasite like malaria. It's actually related to malaria, which means that the typical drugs that are used to treat the bacterial infections will not work against that particular agent. You might have also been hearing about cases of Anaplasma, which is another one of those tick-borne illnesses that can cause high fever symptoms that are similar to Lyme, usually more severely. And then the Poaching Virus, and for some people, and it is a minority, a very small minority, but for some people, the Poaching Virus can cause encephalitis, and that ultimately usually ends up being fatal. And I want, the research is headed toward the idea that if you have one, if you have Borrelia burgdorferi, for example, if you have Lyme disease, and you're infected with Babesia, or if you're infected with Poaching virus, the symptoms are actually more severe because of the kind of relationship that goes on between the microbes that enhance each other's pathogenicities. So that is something of real concern, because nobody actually knows why people with Poaching virus, some develop encephalitis and most don't. So what is the factor that causes the encephalitis? And it might be the presence of one of these other microbes. So Exodes is by far the most common type of tick that is associated with tick-borne illness, but the good old American dog tick, which Dermacenter is the scientific name of that, can transmit Rocky Mountain spotted fever. It can transmit tularemia, which is another very difficult to diagnose and treat type of disease. And lucky us, we, starting with the Long Island and moving north, we are now being invaded by the Lone Star tick, which is amblyoma. The Lone Star tick transmits Ehrlichia and then a disease called starry eye, which is Lyme disease, but it's not caused by Borrelia burgdorferi. The symptoms are basically the same, including the rash, and that's what that stands for, Southern Tick Associated Rash Illness. Also tularemia, and the amblyomma tick is associated with something you may have heard of called a meat allergy, which when you eat meat, you can go into anaphylaxis, which is related to the tick spit, an allergic reaction to the tick spit, but nobody knows exactly why some people get that allergy and others do not. So there's room for plenty of research there. And as I mentioned, Exody Scapularis is the tick most commonly found. And this is a map of New York State that is a surveillance map. So each of the little green dots represents a tick that was submitted to a tick testing laboratory. And in this case, this is a lab called Technology, which I will show you in a second. And when you look at each dot, it tells you the basic location from where the tick came and then what was in the tick. So all of these green dots, the green represents Borrelia burgdorferi, or a Borrelia species, because that they test by looking for the DNA of the organism. So you can see, just kind of looking at our area right in here, we have a wide spattering of things, but by far the most common organism encountered is Borrelia burgdorferi, the cause of Lyme disease, followed by, I think, Babesia microtei, which is the causative agent of babesiosis, which, by the way, is like malaria, can be a long-term chronic illness that comes and goes in terms of symptoms. And then also anaplasma. Anaplasma is a disease that it can be very complicated. Well, mostly just severe, because you end up with very, very high fever and profound illness. So, and if you look at any one of these ticks, like for example, this tick, it's divided in half because in that tick, they found 2 different microorganisms. So you can kind of get a feel for what microbes are found in the ticks that are being sent for testing in New York State. And I bring that up because one of the things that you can do, in addition to knowing who your enemy is, knowing the risk, something else you can do is, not all ticks carry all of these microbes. Many carry 1, others carry 2, and then there are some that carry multiple microbes during the course of their life that they can transmit to you. So, and because of the conflicted medical advice that you might receive when you get a tick bite, what you can do personally is, when you find a tick on you, send it to a laboratory and have it tested to find out what microbes, what pathogens are actually in it. So there are many laboratories, but there are 3 that I think are worth checking out. One is in New York State, and it's for free. That is the Thangamani Lab at Upstate Medical Center. And there's the website, so you can actually go to their website and download the forms you need to send your tick in for testing. This is free for New York State residents. And one of the things that I actually like about all of these tick testing labs is that they give you data. So once again, to assess your own risk, you can go look at the data about what kinds of ticks that were sent in for testing, because they identify the ticks. And then they also will tell you what types of pathogens were identified in the tick. And then they make that data available for every year. So this is actually 2019 data that we're looking at. And I just wanted to show you because I found it very interesting. Notice in New York State, right, the lab is located right here. So that's kind of why the darker colors indicate more ticks have been sent from that area, and that, the word is out there. It hasn't spread too well across the state, which hopefully will be changing. But if you notice, this is the life stage of the tick. The ticks have 3 life stages. The baby ticks, which are the larvae, do not really transmit illness to us because they're too tiny. They don't get on us all that often, to humans that is. Nymph stage do, and then perhaps the most profound, the one that transmits the most, are the adult female Exody ticks. These are the ones that are distinguishable because they have their exoskeleton, their backs, if you will, have a red ring around the bottom of it. They are the ones that we typically run into in the fall months, more so than others. The nymphs are the ones that we run into more in the spring months. Both of them can carry microbes. And just realize that an adult tick has gone through 2 life stages, 2 sets of feedings on hosts, 2 opportunities to pick up more microbes. And so the adult ticks are the ones that typically are infected with more than one microbe. And then, as I mentioned, notice there were several types of ticks that were discovered, but Exody Scapularis, which is the black-legged tick, is the one that was found most often. And, you know, I just want to, this one, Exody Scapularis, is something called the woodchuck tick. And they are here, so, and they can transmit diseases. So rarely, but nonetheless. And all of these ticks, by the way, unless you are a tick expert, are very difficult to tell apart. So realize ticks are arachnids. They have 8 legs. So if you find something on you that has 6 legs walking around, it's probably some kind of other insect. Ticks are arachnids. So check out the number of legs. If there are 8 of them, you need to be concerned. Unless, of course, it's a spider, which you probably should be concerned about anyway. Looking at pathogens, and again, by far the most prevalent of them is the Borrelia burgdorferi, which is the cause of Lyme, followed by, interestingly enough, Ehrlichia, and then also Babesia. So in our area, again, notice this is sort of more centrally focused, but in our area, and one thing about these maps is they're interactive, so I could actually go right to Warren County, for example, and it would tell me exactly what percentage of ticks had what. So that's one of the real big advantages of this location. So you can keep track of the data, not quite real time, but very close. They were closed during COVID, but they are reopened. And again, the tick testing is part of a research project. So your tick becomes part of a research project. So you're adding data to the database to help protect other people as well. So this one is our local lab. Then I just wanna, tick report I've had huge success with. They are excellent. They're associated with UMass at Amherst, which is our next door neighbor in Massachusetts. At tickreport.com, this is not free, so you have to pay for it, but for the cost of the test, you have results back from 8 different pathogens. So it's a very good opportunity to have more data. And again, they provide the data publicly. So on their website, they will show you all this data. So I just grabbed a snapshot of some of the kinds of things that you can find out, mostly because I found this and this very interesting. So the tick bite subject age, right? So notice that kids, right, certainly get their share of ticks. Teenagers are playing video games, so they're not so much. But then notice it goes up as the grown-ups get out in their yards doing yard work. We're the ones walking the dogs. We're the ones doing those kinds of things. And so, you know, the 40 to 50 age group is the one that actually has the highest number of ticks. So remember, this is biased because also, you got to wonder if a teenager finds a tick on them, if they're going to be like, Mom, I got a tick, send it in. As opposed to adults who we've realized at this stage that we've got something to be concerned about. So we would be more likely to do that. And then in terms of this, where on the human body are ticks found? And notice it's pretty evenly distributed across the body. So, you know, everybody says, check the back of your knees, check here, check there. You know, if anything, the one place that comes up is the upper leg. And the rest is just any part of your body is susceptible to tick bite, because that's what they do. They latch onto you and they start moving around, they start climbing, they get under your clothes till they find a good spot, whatever that is for them, and they attach and then start to feed at that point. But there is no one spot, which is why tick checks are so important. And you need to check everywhere when you do those tick checks to make sure that they are not in places that you normally wouldn't look, right? A quick look on your leg, a quick look on your arms is not going to be enough to protect you from transmission of disease. So that's lab number 2. And then lab number 3 is technology, also excellent. When the other labs were not working, this one actually was. Coverts, in other words, they were accepting samples throughout the whole period. So again, more data, and this is a point that I wanted to make, which is very important. Most people feel that tick season is spring. That's when the nymph ticks are feeding. And there has been a bias in the scientific world that the most important tick, the one we have to worry about the most, is the nymph stage tick. So that's not true. So the nymph stage tick, this peak you see right here, that would be the time in May, right? May through early July, where the nymph ticks are out feeding. We come in contact with them. And I also want to point out during this early period, any of the adult ticks that did not feed in the fall, winter over, wake up and wake up hungry. So this is a mixed bag right here of adult ticks and nymph stage ticks. So the teenagers and the adults are both feeding during this period. So your risk is probably highest, but I also want to show you this other peak that, end of September, as the temperatures become cooler again, those ticks come back out, because really the only thing that they do not like is hot, dry weather. So as the temperatures go back down, anybody who didn't get breakfast during the early feeding season will be feeding during this period here. And that this data shows that very, very clearly. So I guess the main point to make is that you are at risk of getting a tick-borne disease pretty much any time of the year here. And when you go to technology, they actually have an interactive map showing, day by day when the tick bites are coming in, when they're getting tick submissions and what's in them. And that is very interesting to look at. I would like to show that to you, but I cannot share that part of the screen. So go to technology.org, go to New York State tick submissions, and watch that interactive map so you can see that your risk of getting a tick bite at least is highest. And as far as preventing a tick-borne illness, at the time of the tick bite is your best chance to not have to deal with any kind of tick-borne illness, because if you see the tick and remove it, the earlier you can remove it, the more likely you will be to be safe from that tick. So the ticks do have to be attached for a period of time. It's far less than 36 hours. The data that resulted in that conclusion, the scientific publication that said that, and there was like one of them, was not based on human data. It was based on mice, and it was all done with nymph stage ticks. So the point, all we know is that nymph stage ticks take longer to transmit disease. Adults probably can transmit in less time, mostly because they're bigger and because they have done this twice. So just realize that, that 36 hours and they have to be engorged does not mean you are safe. If you find an engorged tick, that is one that you really want to be concerned about. But even if you find an attached tick, and by that I mean if you have to work to get it off you using tweezers or something else, then that you are at risk of having gotten a tick-borne illness. So this is a paper that was published, just a little while ago, which I thought was very interesting because it shows how much Lyme disease, and this is kind of focusing on Lyme disease, has expanded since 2010. This is a map of positive lab test results, and I'll talk about those lab tests in a second, but nonetheless, so looking at all the data from Quest Laboratories, in 2010, notice, so the lab tests, the positive lab tests were kind of centered in the Northeast and here in the upper Midwest, which are the spots where Exody Scapularis, the deer tick, the black-legged tick, is usually found. And as awareness of Lyme disease has increased, the number of tests have increased overall. And notice the proportion of positive test results have increased. So from 2010 to 2016, six years, notice basically there are positive lab test results from across the state, everywhere except for South Dakota, which is kind of interesting because South Dakota also happened to be one of the states that didn't end up with a whole lot of COVID in it. So maybe they have superpowers, but nonetheless, notice how much Lyme disease has spread. This is an epidemiological issue. So this is no longer an issue that just affects the Northeast. This is a national issue that requires or should alert people to the fact that we need a national response to this. And that is slowly starting, but still it's nowhere near what it needs to be to get a handle on this disease. So this is a very interesting paper. And if you're interested, all you have to do, you can search for this title. And it is open access, so that means you can read the whole paper if you want to. Lots of interesting data there too, for those of you who like data like me. So all of the above. You miss the tick, you find it later, it's engorged. What are the symptoms of a tick-borne illness? So what you need to realize is that the symptoms are very similar to other diseases. The early symptoms of Lyme disease, of actually all of those, are very similar to other infections. And that would include COVID, that would include West Nile virus, that would include influenza, that would include other viral or bacterial illnesses. Even the tick-borne illnesses in those early stages are similar to each other. So it's not always easy to make a diagnosis. And again, that speaks to how bad the lab tests for Lyme disease in particular are. So notice that this is from the CDC. So I actually just went and borrowed it from them in terms of what are your symptoms. If you were bitten by a tick and develop an illness, what are the symptoms? Notice whether or not you got the disease varies by geographic region, depending on what ticks are present in that region and what diseases they carry. And notice the symptoms: fever, chills, aches, and pains. So those are the early symptoms of pretty much all of the tick-borne illnesses, and also many other types of diseases. Difference being, if it's a virus, there are no real antiviral types of therapies for it. It's just something you have to ride out. If it's a bacterial infection, if it's Anaplasma, if it's Lyme disease, if it's Babesia, medication, right, antibiotics are required as a treatment for that. So it's kind of important to be able to diagnose your disease appropriately. Here's a hint. Typically, viruses like, for example, cold, the flu, all those are not in this area, are less of a risk in the summer. And if you end up with a flu-like illness in the summer, right, they call that summer flu, it is probably not the flu. It is not a respiratory illness that you picked up. It is more likely at that stage that you have a tick-borne or a vector-borne illness, vector meaning blood-sucking insects, because that's when the blood-sucking insects are out and around. So be alert to those symptoms and realize that, it could be something more than just a virus, go home and sleep it off kind of thing. So when you also look at that, this list, aches and pains, and this is a list of those early symptoms, right? Fatigue, headache, rash, fever. The rash has been an issue for a long time because the early research on Lyme disease focused on the rash. The early researchers found this very unique bullseye appearing rash associated with this illness called Lyme, that they ended up calling Lyme disease, which was actually the first patient showed up with symptoms of like a rheumatoid arthritis kind of thing. And so some of those patients had rashes, and so the research focused on people with the rash. So the rate of that rash in patients was biased, right? So it was increased as a result of the focus on it back in the early days. So the rash does not occur in 70 to 80% of the cases. It does not even occur in 50% of the cases. So the bullseye rash. So the going thought about rashes are, especially for the Lyme disease rash, right here it is, less than 70%. It's actually less than 50%. So 50% of people who have Lyme disease, and this has been researched, end up with no rash at all. So the rash is a skin manifestation of the infection only, because sometimes the bacteria go other places. And then of those that do have a rash, usually it's just a red raised rash that gets to be large. That's the most common manifestation. They call that atypical rash because everybody's looking for the bullseye. Just be aware, it does not need to be a bullseye. The most common is the red raised rash. So, and then the bullseye rash, the research actually indicates that good old bullseye, that if you look it up on the internet, the first thing that pops up are 4,000 pictures of a bullseye. That is actually occurring in less than 10% of cases. So it's very unique and very distinctive. And if you get it, I call you the lucky winner of the bullseye, because that's recognizable and that's something that will get treated, as opposed to these other types of rashes that could be spider bites, that could be bites by other insects. It could be an allergic reaction to a mosquito bite. So, it's very hard to diagnose any illness on the basis of a rash alone. So Lyme disease is one of those that the rash has, created controversy in, but notice these other diseases, right? Starry eye, Rocky Mountain spotted fever, ehrlichiosis, tularemia can also cause rashes. So again, going back to how hard it is to actually diagnose early infection, early disease, because that rash doesn't show up most of the time. And the other symptoms are pretty nonspecific. So what is here, this description of tick-borne illness defines what is referred to as early Lyme. It's one of the first stages. Right after the bacteria get into you, you may develop these symptoms, or you may not. And that makes for difficulties when it comes to diagnosis. I also want to point out there is no laboratory test that will be able to tell you whether you were infected early in the disease, like within the 4 to 6 weeks. The serological tests for Lyme disease are typically negative. So that creates quite a problem in being able to diagnose this. Early in the disease is treatable with antibiotics. In fact, that's your best outcome, because 80 to 90% of people who are diagnosed within the first 2 to 3 weeks who receive a standard antibiotic treatment recover fully. 10 to 20% continue to have symptoms. And if you are not diagnosed early, if the rash didn't occur, if the flu-like symptoms didn't happen, if you didn't see the tick, even if you did see the tick and your doctor said, go home, it's probably the flu, the problem with that is it allows the bacteria to spread, disseminate, and then they become tolerant to antibiotics. So it becomes much harder to treat those symptoms. And you'll notice the symptoms morph into, not flu-like symptoms, but they develop into more of a chronic illness type of symptoms at that point. So this goes by various names: chronic Lyme disease, persistent symptoms of Lyme disease, and for one subset anyway, post-treatment Lyme disease, because as I mentioned, even 10 to 20% of people who are promptly treated appropriately still end up with long-term problems. So looking at this paper was published last year, 2019. This is published from the Lyme Disease Research Center at Johns Hopkins University. Dr. John Alcott is the head of that, who has been doing an outstanding job leading the way to try to define what exactly is going on, try to define the patient population of people who have these long-term continuing symptoms. So the post-treatment word, the term actually comes from the fact that, the appropriately diagnosed, treated with 3 weeks of doxycycline standard treatment, that group that still continues to have symptoms, that's the group that really represents post-treatment Lyme disease. So who's left out of that are people like my daughter and many other people who were not diagnosed early. The bacteria were allowed to disseminate. Then they become treatment resistant. And by treatment, I mean antibiotic treatment. They are resistant bacteria, naturally resistant. So the disease becomes much more difficult to diagnose. So what they've shown is that so rather than call it chronic, they're calling it persistent symptoms of Lyme disease. So they're basically saying that post-treatment Lyme disease is a model for this other group of people like my daughter, which I think is excellent. And I'm very happy that they are doing this. They're characterizing those patients so that they can kind of be able to distinguish them. And this was a chart from the paper. Notice that looking at the number one complaint is fatigue. 100% of patients with post-treatment, or I'm going to call it chronic Lyme, with chronic Lyme, have fatigue. And I'm not talking about gee, I'm tired, I worked really hard today, I'm really tired, I'm going to go to bed. I'm talking about bone crushing. If the house was on fire, I'm not getting out of bed to get out of it. Fatigue, profound fatigue, joint pain, and then a good deal of neurological issues are also included in that. So this validates that this is a true disease state, that It isn't an exaggeration. It isn't somebody's mental illness. It is actually a biological and physiological problem. And it is. And the problem with this is it's still not very well accepted that this occurs, again, for reasons that anytime you want to hear the backstory about how we got to where we are today, I'd be happy to share that too. But the point being, it is a real biological condition. And now what we need to do, it's treatment resistant, antibiotic treatment resistant at this stage. We need to figure out why does it occur? Is it because the bacteria are still there causing issues, or is it because your immune system has been made wacky as a result of the infection? That research is going on. But the validation that this actually occurs is quite profound in this field. So that persistent symptoms of Lyme, I call that the elephant in the room, because it truly is. This is where so many Lyme disease patients slip through the crack. This is the story of my daughter. This is the bacteria were allowed to spread, and then antibiotics, helped, but did not return her to health. She still had a significant number of neurological symptoms that went on for a time until we were able to identify a treatment that was actually several approaches that were actually able to return her to good health. So as I showed you on the previous slide, this is a real biological disease. It affects the physiology, the human physiology. There are symptoms that are obviously more common in the patients than in the controls. So on the other side of that, there have been a large number of research teams that have been trying to figure out what it is that is causing these symptoms. Is it because the bacteria are still present, or is it because, as I mentioned, the immune system has gone completely wacky and is now attacking your own tissues? That's called autoimmune disease. So there's sort of a war between those two factions, because think about it, the treatments, they're two different specialty areas. If it's the bacteria that are still there, it's an infectious disease issue. If it is an autoimmune disease, then you go to see a specialist, a rheumatologist. And the treatments are different. So if it's an infection, you would expect to get antibiotics. If it is an autoimmune disease, you would expect to get steroids to decrease the inflammation and turn down the immune response. So we need to know what that is for obvious reasons to improve treatment outcomes. So what we know on the biological side, and this is where I hope not to become too microbiological for you, because I can't help myself here, because that's what I do. But we know that, and how do we know this? We know this from in vitro test tube studies. We know this from animal studies, several different types of animals, and most profound, we know it from non-human primate studies. Non-human primates are substitutes for humans. Why? Because you can't infect a human with a disease and watch it linearly to see what the outcomes are. We did that with syphilis very unethically, and that is not something that we do anymore. So you can watch those patients that are infected and see how they respond to treatment. But with non-human primates, you can infect them and then follow the course of the disease, and then at the end see what the pathology is, right? So actually do a pathological study to find out, are the bacteria still there or are they not? So from all those different studies, what we know is that Borrelia burgdorferi, and actually all of the different species of Borrelia, they are spirochetes. They are remarkable bacteria. Even in the realm of all bacteria, these are perhaps the most biologically remarkable in terms of their being able to, what they're able to do to their hosts. That would be us, but mostly their hosts are animals, right? So we just happen to be the spillover host. But they have the ability to talk to our immune systems and say, nope, stand down, here we are, but we're fine, don't bother us. So they can escape from those early immune responses that are designed to go find and attack bacteria. They get away from those things. They get into the bloodstream because they're corkscrew shaped and they can drill through tissues to get in there. They are carried through the blood to their target tissue, and that varies by species what target tissue they prefer. usually the heart, the joints, or nervous tissue. And then they start to colonize. So they reproduce, and then they actually change biological forms. They become less active. They become a community where they are talking to each other and responding to each other. It's something called a biofilm. And they establish those colonies in those preferred spots, and they just start to grow. It is at that point that antibiotics are no longer effective because they become antibiotic tolerant, which is, microbiologically speaking, different than antibiotic resistant, that they tolerate antibiotics. They could care less if you take antibiotics. They're not affected by those antibiotics because they're not in the same metabolic state as they were when they entered your body. So they call those persisters. I refer to them as persistent forms. Resist antibiotics or tolerate antibiotics. So once those colonies are established, now there's a bigger problem, because the colonies trigger inflammation, which is part of the early immune response. It's what our bodies are supposed to do, but our bodies are also supposed to turn it off when mission is accomplished. Mission is not accomplished with these types of bacteria. So the inflammation increases, and inflammation breeds more inflammation. So it increases, along with other immune system changes that go along with that. We also know by immune system is altered, we know this at this stage, that Borrelia burgdorferi suppresses the overall immune response in a way similar to how other better known pathogens are able to suppress the immune response. So they go right into our lymph nodes and basically tell one set of cells to stop talking to the other set of cells, turn this on, turn that off. They're in control of that response. So that means the immune system is impaired, it's altered. So you have, increasing inflammation, and you have decreasing ability to deal not just with the Borrelia infection, but other infections as well. So that's where the concept of co-infections comes in. Bacteria that would normally not be an issue, like Bartonella, like Mycoplasma, like chlamydia, take advantage of that situation of suppressed immunity and just have fun with your body. So the problem becomes not just the one agent, but now you have other secondary infections too that are equally difficult to treat. This is something that is seen with HIV. It is undisputed with HIV. It should be undisputed with Lyme disease, because we have the data, we have the evidence that that's what's actually going on. So it's still an elephant in a room, because if you talk to an infectious disease doc, they will say this doesn't happen, this doesn't occur. And if you talk to other docs, they will tell you, my patients, this is real, and we need to do something about it. And, that's kind of the grim perspective. And so I just realized the landscape is changing. When my daughter first became sick in 2010, we had, it was Wild West in terms of the research was, it was there, but it wasn't respected, because the voices that were saying, this is no big deal, Lyme disease, simple bacterial infection, it's hard to catch, it's easy to cure. Those voices were significantly louder, but, as the patient population grew and more people were affected by this disease, the landscape is changing. And that continues. It has been a slow and steady march, but for those of you who might understand science this way, science works that way, right? So it takes time for standing principles for the science to accumulate that says, oh, that was wrong and you need to change your mind. Plus you have a bunch of people who are very deeply invested in that whole concept that, no, we got it right back in 1980. And are having a hard time realizing that, well, maybe you didn't get it 100% right. So here are some of the things that have happened in the past 8 years anyway, thanks to legislation that was initiated by Congressman Chris Gibson when he was here in our district. The legislation to create a tick-borne disease working group was incorporated into the 21st Century Cures Act, and the Department of Health and Human Services convened the first tick-borne disease working group in 2018. So the 2018 report to Congress is available to read in full, or you can read the summary, or you can read even the individual subcommittee reports if you want to. I was a member of the testing and diagnostics subcommittees, so you can kind of see it lays out what are the problems, what are the potential solutions, where is the science lacking. It's a very comprehensive report, and I'm proud to have been a part of it. So as a result of that, NIH came up with a strategic plan, basically saying we need better testing, which I'll get to in just a second. We need better testing for this disease because at the moment, there is no way to diagnose Lyme infection early in the disease when it's treatable. There is no way to diagnose, there's no way to determine what chronic Lyme disease is by laboratory test. So all of those are needed, and NIH at this point realizes it and has made it a priority to move forward diagnostic tests for Lyme disease in particular. So that on the legislative front, on the advocacy side, which is what I have worked on both at the federal level and at the New York State level. And then on the research front, all these within the past 3 years, the science has advanced significantly. And that is because philanthropic groups, such as Focus on Lyme, but others, are funding the research that our federal government should be funding. So the funding for Lyme disease is actually very small compared to the funding for other types of diseases, even infectious diseases. So the Lyme community has stepped up and is funding research now. So I am the scientific advisor for Focus on Lyme. Focus on Lyme is based in Arizona, but we, in my role as a professor of microbiology at SUNY Adirondack, we are collaborating with them on a research project, on actually several research projects that Focus on Lyme is funding. So one in particular has led to a lot of very exciting details about Lyme disease in general, but the most important thing is that from the research we've been able to identify a group of peptides from Borrelia that will be able to use as part of a diagnostic test. It is at the point where before the end of the year, we hope that we will be able to begin clinical trials on that. It is highly accurate. It is accurate in the earliest stages within a week of infection. So if you go to the doctor with a tick attached and the doctor says, you just gotta wait and see if a rash shows up, which is that was the message that we got, you will not have to do that anymore because there will be a test available to tell you whether you were infected with Borrelia burgdorferi or not, and you can start antibiotic treatment immediately, which would reduce the rate of chronic illness associated significantly. So that is very exciting. And I also want to point out that the patient data that Johns Hopkins has been accumulating for post-treatment Lyme is validated with this technology called immunosignature. So there is a definite immune signature that corresponds with those with persistent symptoms of Lyme disease. And now that needs to be parsed to figure out what exactly is it the immune system is responding to. Is it Borrelia, which for many it is, but for other subgroups, it appears to be something else consistently. And that is where we're going next. So look for that within the next year. And those of you who participated in our biorepository project, in other words, you showed up and you rolled up your sleeve and gave blood for that, that's you. You did this. It was those samples that allowed us to fund this research, because that's one of the big problems with Lyme disease. There are very few biorepositories. And now we have very good data. We're working with Arizona State University, the Biodesign Institute, the Institute for Biodesign researchers, and very good news there. I also want to point out Duke University School of Medicine has developed, working with Dr. Neil Spector, who very, very sadly passed away. We are, much of the research being done now is being dedicated to him because of the hard work that he put in. He is the Duke University oncologist who years ago developed Lyme disease that went to his heart, caused Lyme carditis, which went undiagnosed for 10 years. And at the end of it, he had to have a heart transplant. When they removed his heart, they found spirochetes in the tissue. So the spirochetes caused his heart disease, which led to his heart transplant. So he lived successfully with a heart transplant for 12 years, but this past year, because of the immunosuppressive medication he's been taking for it, he battled with several types of cancer and ultimately lost his life to that. And that was just last month. It was a huge loss to the community. And he was an amazing human, probably the most amazing human I've ever met. But his research continues with his colleague, Timothy Hayward, and what they have found, based on their experience with cancer research, they found a targeted therapy that will allow for the destruction of Borrelia burgdorferi wherever it happens to be in the body, meaning it will go to those places in those tissues where the antibiotics won't go, bind, deliver a toxin, just like with cancer, just like those therapies that they're now using successfully for cancer. And the good news is that could also become a diagnostic. So it could be an imaging approach. So you get diagnosed by this immunosignature with persistent symptoms of Lyme or chronic Lyme disease. And then you go have that imaging test and says, yep, the spirochetes are right there in the joints. Now we deliver the toxic drug. So these are both very, very promising things. For the first time, it looks like, you know, we have a way to approach that disease, that form of the disease. And then also just a shout out to the Tulane University National Primate Center. Dr. Monica Embers has done most of the biological research that has allowed us to figure out what those spirochetes are up to and just how tricky they are. And also that, you know, persistent Lyme, for at least some of them, some people also never produce an immune response to Borrelia because of the ability of those bacteria to completely subvert the response to it. So it's called seronegative Lyme disease. And it means that you're going to be negative for those serological tests because you're not producing those antibodies. Because why? The bacteria told you not to produce those antibodies. So she was able to show us that. And then also, as I mentioned, the Johns Hopkins Lyme Disease Research Center is doing has the largest body of clinical patients, both acute and chronic patients, that they are busy delving deep into what is going on with the patients. So in other words, the test tube research, and now we have patient data and biomarkers, you know, all kinds of different biological data coming from the clinical research side of it all. So from these areas has emerged you know, like I said, I'm very hopeful that within a year or two, this is going to be less of a problem. And, you know, NIH has already, you know, expressed interest in moving this further. So I'm actually very optimistic, a little less grumpy than I might have been in previous years, since this is my third year doing one of these talks. So if you are interested in more info, feel free to go to any of the websites that I've shown you. Here's our website, right? Lyme Action Network is local. We are here, right here in the eastern part of New York State. And then Focus on Lyme, again, the organization, we are actively funding research. So a lot of what we do is fundraising so that we can say, you know what, we want you to do this research because this is why. It's going to tell you what to do. They funded the biorepository. And then also, just for basic information that's accurate, those three organizations, LymeDisease.org, Global Lyme Alliance, which is also based in New York, and Project Lyme. There's also Bay Area Lyme, which is, they have a biorepository of acute samples that you could go to. But all of these have good, accurate information that you can send people to if they have questions or if they want to learn more about Lyme disease, the risks, and also what to expect if you are infected. So I hope that you will take advantage of those, and I hope that you enjoyed this talk, and I will be expecting questions at the end of all this. So, and you can always reach out to me. I am at SUNY Adirondack, so you can certainly reach out to me directly if you have any questions or thoughts or concern. So thank you to the Lake George Land Conservancy for inviting me to give this talk. I hope it was helpful to you, and looking forward to talking with you more later. Thank you.